thank you for joining the webinar today. I am Sarah Traynor. I'm a research scientist at Active Motif, and today I'll be talking about recent technology advances in epigenetic profiling, really focusing on three key methods that are offered here at Active Motif, either through kits or end-to-end services. So let's get into it. As I said, I'll be covering three different genomic profiling methods. I'll be giving a brief introduction into all three of them. So I'll be starting with ChIP-Seq, moving into CUT&Tag, and then CUT&RUN. I'll be providing advantages and considerations of each method. And then I'm going to get really into comparing each method, when to use each one, and giving you some insight there. So let's get into what is ChIP-Seq. Well, ChIP-Seq is the gold standard method that is used for mapping protein-DNA interactions and histone modifications across the genome. It's a technique that combines chromatin immunoprecipitation with next-generation sequencing, and it is used to identify binding sites of DNA-associated proteins throughout the genome. Typically, you do need to use some sort of cross-linking agent. Formaldehyde is commonly used, and the reason for that is you really want to lock in those proteins at those DNA binding sites. So just to briefly go over the standard workflow here, the first step is a cross-linking step, again, typically using formaldehyde. The next step after that is cell membrane lysis, and then after that, we have a fragmentation step. This is typically done using a physical fragmentation like sonication, but you can use an enzymatic fragmentation here as well. The following step is the immunoprecipitation. So this is really the ChIP in ChIP-Seq. You use an antibody that targets your protein of interest. You're going to incubate your chromatin with that antibody and really enrich or isolate the DNA sequence that is bound to that protein. After that, you have a reverse cross-linking step, and then followed by library preparation in order to get those sequences ready for sequencing analysis. Moving on to the next method, which is cleavage under targets and tagmentation, otherwise known as CUT&Tag. It's a method that was first introduced by Dr. Stephen Hennikov. It's a rapid and robust method for genome-wide analysis of histone modifications, and it is specifically useful at lower sequencing depths. It's a method that uses an antibody tethering strategy in order to target specific histone modifications and some transcription factors. So the antibody is used to target your histone modification of interest. And then what is actually tethered is an enzyme called protein A TN5. So this is a TN5 enzyme that's fused to protein A. And the protein A portion is going to bind to the antibody that you are using to target your histone modification of interest. So to go over the standard protocol here, you have cells that are immobilized onto concanavalin A beads, and this is just used to make the wash steps easier. So your first incubation is with your target-specific antibody. You're going to sequentially add a secondary antibody, and this is just in order to recruit more PATN5 to the target site. After that, you're going to add your protein A TN5. Again, the protein A portion of this enzyme will bind your antibody that's targeting your protein of interest. And then you can activate the tagmentation using magnesium at 37 degrees. And what this is going to do is it's going to activate targeted tagmentation. So you're only getting the tagmentation at that specific site. You can see you're getting the cut and release of the sequences surrounding your protein of interest. After that, you extract the DNA, and because we are working with TN5, this means that no secondary library prep is required. So all that's required is PCR amplification of the extracted DNA, and it is ready for sequencing analysis. The next technique is cleavage under targets and release using nuclease, otherwise known as CUT&RUN. CUT&RUN is another method that was also introduced by Dr. Stephen Henikoff, and it enables sensitive and specific genome-wide analysis of chromatin-associated proteins. This is a primary antibody-based tethering strategy to target protein DNA complexes. And like CUT&Tag, again, there is a targeted strategy here. However, rather than using PATN5, it is a protein A, protein G mycococcal nuclease that's used, otherwise known as PAG eminase. And this is what's actually recruited to the targets and is going to do the specific release. So again, let's just go over briefly the standard workflow here. Again, you have cells that are bound to concanavalin A beads, once again, just to make the washes easy throughout the protocol. The first incubation is with your target-specific antibody. So this method does only use a primary antibody. There's no need for a secondary antibody here. So the next step is incubation with your PAG eminase. Again, the protein A, protein G portion of this protein is going to bind to the primary antibody and be recruited to your target site. Upon activation with calcium, you get the targeted release, cut and release of the DNA sequences that surround your protein of interest. Again, you release the DNA, and now because we are not working with TN5, we're working with MNAs, you will need to do a secondary library preparation in order to prepare your fragments for sequencing. So here I have genome browser tracks that were all generated using K562 cells and the H3K4 trimethylation histone mark. The tracks were generated either using ChIP-Seq in pink, CUT&Tag in green, or CUT&RUN in blue. I've also included an ATAC-seq track, but I'll talk about that in a little bit. The first thing I want you to notice is the similarity between the CUT&RUN, CUT&Tag, and ChIP-Seq tracks. As you can see in both CUT&RUN and CUT&Tag, you do see slightly less background, but the important factor here is that you are getting tracks that look very similar to the gold standard, which is ChIP-Seq. A key benefit, and I will talk about this more in a few slides, is the input amount that is used for CUT&Tag and CUT&RUN. It is much lower than what is used for ChIP-Seq, so you're getting very similar data with less input. So now to get to the ATAC-seq track, the reason I've included this is specifically for CUT&Tag. So in CUT&Tag, like I've said, we are using PATN5, and so a question amongst researchers may be, what about free PATN5 in the reaction? And that is a valid question, and this is the reason I did include this ATAC-seq track. And so as you can see, there are some overlapping peaks between H3K4 trimethyl and the ATAC-seq track. You would expect this. H3K4 trimethyl is going to be in open regions of the chromatin. What you also see, though, is you see some peaks that are seen in the ATAC-seq track that are not seen in the CUT&Tag. The reason for this is the conditions in the binding buffer don't actually allow free PATN5 to bind to open regions of the chromatin and tag there. Instead, all the tagging is going to occur specifically at your region of interest where your antibodies are. Next, I'm going to talk about the advantages and considerations of ChIP-Seq. The advantages include the wide compatibility with a wide range of sample input, including tissue, as well as a wide range of targets, including both histone modifications and transcription factors. However, there are limitations to this method. One of the biggest limitations is the amount of sample that's required. This method does require a large amount of cells or tissue, whatever your input may be. There's also equipment required for sonication, and a separate library preparation kit is required since the fragments that you're pulling down in your immunoprecipitation don't already have the sequencing adapters. Overall, it is a complex workflow. It's quite laborious and time-consuming. And then the other big limiting factor is the sequencing depth. It does require a high sequencing depth, which adds cost to each experiment. These limitations are really what prompted the need for other methods capable of mapping protein-DNA interactions throughout the genome, and it is why we now have methods like CUT&Tag and CUT&RUN. So going on to the advantages and considerations of CUT&Tag. First of all of the cells remain in the native state, so no fixation is required like it is with ChIP-Seq. However, it is still compatible with a light fixation. It's a simplified workflow, so it does reduce hands-on time. CUT&Tag can be completed within 1 to 2 days, so it is a really quick protocol. One of the biggest benefits is the low sample input. It's quite significantly lower than the input that's required for ChIP-Seq, so that is a big advantage. No secondary library prep is required. This is because, again, we're working with PATN5, which means the sequencing adapters are inserted into the DNA fragments during tagmentation. So all that needs to happen after the fragments are isolated is PCR amplification. Decreased sequencing depth. This, of course, translates to a lower cost per sample. So that is a huge advantage of this method as well. Limitations here is the primary compatibility with histone modifications. This method, like I said, it does work with some transcription factors, but really you're targeting histone modifications if you're using this method. Next, we have the advantages and considerations of CUT&RUN. So once again, these cells remain in the native state. So no fixations required, but again, it is compatible with a light fixation. It requires only a primary antibody. So this further simplifies an already simple protocol. Again, this only takes 1 to 2 days to complete. So it is quite a simple workflow that reduces hands-on time. It has a wide range of targets, including transcription factors. So CUT&RUN can be used for both histone modifications and transcription factors. And again, we have a decreased sequencing depth that translates to a lower cost per sample. Now, the limitations here are that a separate library preparation is required. This is because you do not have the sequencing adapters on your fragments, so you need to ligate those on afterwards. Now, I did put restricted to 500,000 cells in the limitations because it is a limitation. We are telling you how many cells you need per reaction. However, what I do want to point out is this is still lower than what is required for ChIP, especially if you're looking at transcription factors. With all that said, let's move on to comparing these methods, and then I can give you some insight as to which method you should select in some scenarios. So here we have a table of each technique, and we have some critical considerations when deciding which method you should use. First, can this be performed under native conditions? Is a chromatin fragmentation method required? What are the cell requirements? What's the sequencing depth? Is there an integrated library preparation? What are the compatible targets? And what about the workflow length? So in a case where you're not limited by the sample amount or the sequencing costs, and you're comfortable with a more complex workflow, you have 2 to 3 days to complete the assay, we're always going to say go with ChIP-Seq. ChIP-Seq, again, it's the gold standard. It works very well, but you do need 1 to 10 million cells. So this is quite a lot of material that is required to perform this assay. So that's something to keep in mind. In addition to that, 20 to 50 million reads. This is quite a deep sequencing depth for this assay. So if you're not limited by these factors, go with ChIP-Seq. In a case where you do have limited sample amount or tissue and you're targeting histone modifications and you want to limit the sequencing costs, CUT&Tag is what you're going to go with. So this is a great assay for targeting histone modifications. It only requires 5,000 to 500,000 cells. So that is much less than what is required for ChIP-Seq, and you only need 2 to 8 million reads. So that is going to be a lower sequencing cost per sample. Another thing to keep in mind with this method is the simplified workflow. So it only takes 1 to 2 days. So if that is something you're looking for, that is another reason to select CUT&Tag. In the case where, again, you're trying to reduce the sequencing costs and you have a limited amount of sample, but you're targeting transcription factors, we're going to say go with CUT&RUN. So yes, this does require 500,000 cells, but again, that is less input material than what is needed for ChIP-Seq. And in this case, only 8 million reads is required. So another reason to choose this is limiting sequencing costs. Another thing to keep in mind is, again, the simplified workflow, only 1 to 2 days. So a simplified workflow, limiting the sequencing cost, and if you're trying to conserve the amount of sample you have, go with CUT&RUN for transcription factors. Now, with all that being said, I just want to wrap up here and highlight that here at Active Motif, we do offer many different end-to-end services. We're not just limited to the 3 that I covered today, as well as we have many different types of kits and products to offer as well. We have tech support available if you want to refer to our website. We also have many resources there that will ensure that you are choosing the right method for your investigation. So I'm going to pause on this slide for just a few minutes. If you are thinking of doing any ChIP experiments, scanning this QR code will take you to our ChIP selection guide that will walk you through which ChIP is right for you. And if you want any more information on the methods I discussed here today, ChIP-Seq, CUT&Tag, or CUT&RUN, here are the links to our website. Like I said, there are many resources on our website, a lot of good stuff that is going to help researchers like you. Thanks for listening, and I hope it was helpful.